Um, so it gives me great pleasure to welcome Daniel Green. Daniel is the Kenya Field Program Director at the Department of Human Evolutionary Biology at Harvard University. And as fate would have it, he was coming through Vienna on personal business on his way to Mainz to work at the Max Planck Institute for Chemistry. And so we took the opportunity to offer him a talk slot, and we're really delighted that he's here. So I'd like to welcome Daniel to give this occasional HEAS talk on dental records and the origins of the modern African world. And hopefully, everyone, you can hear me okay, and just give us a yes in the text if you can, just to confirm once and for all that that we are live on online. Thank you.
Thank you so much, Tom. Oh, great. People can hear. And thank you, everybody, for waiting. I wish I could promise you that this was going to be worth the wait, but in any event,
because I left the key.
thank you so much, Tom and Dave and others for hosting me. I'm really pleased to be able to be here at your wonderful center and to be meeting some of the really brilliant it's been spectacular. So all of you work in the past in some way, and there are so many different questions you can try to ask and answer with many different methods. I'm going to be trying to ask a little bit today about where the African world that we see today comes from, specifically focusing on dental records. And as I'll try to argue in this talk, while there are many different ways we can look at the past, because teeth are so ubiquitous in many terrestrial sediments and marine sediments as well, and because of the specific way that they form with this kind of interesting mineral and protein-based interaction, they really contain a lot of interesting and important information about the past. So just to move forward and let me know, people on Zoom, if you can't hear, I'll try my best to stay close to the computer. I just want to begin by saying this work really depends upon quite a few people. In particular, I want to thank collaborators from the Turkana Miocene project, many of whom are listed here. We have over 60 collaborators from over a dozen different institutions. Also, some of the data that I'll show here today come from a number of people outside of that collaboration, in particular, Tanya Smith, Ian Williams, Tom Cleland. I just want to mention their names. And I'll come back to some of them later. And I also want to thank some of my funding sources, including the Turkana Basin Institute, which is based in Kenya, which has been really crucial, the National Museums of Kenya, which have made these specimens and data collecting possible, and the National Science Foundation as well. So today I'll speak about a few things. I'm going to take us through the Cenozoic, focusing, of course, on Africa, and I'll try to make some case that the patterns that we see in Eastern Africa, and in the Rift in particular, mirror and also differ from some of the patterns that we see in the larger global records. I'll then try to zoom in a little bit on individual behavior of different kinds of taxa, and in particular on primates, which are quite important for our own ancestry. And one of the reasons why we can even look at that kind of information of individual seasonal records from the teeth of specific fossils is because of this kind of interesting, again, inorganic-organic interaction in the formation of teeth, which leads us onto the question of proteomics. And that can be interesting because we want to reconstruct, for instance, climatic records on a seasonal basis in the past, but it also might be useful because we're interested in the proteins themselves, and I'll get to that at the end of the. So to start with the Cenozoic, many of you will be familiar with a curve like this, either from the Zachos, famous 2001 Zachos paper, or a more recent iteration of Westerhold, from which I'm deriving this data. But basically, you can see on the y-axis on the right some kind of variation in temperature over time on the x-axis, and this is derived from oxygen isotope records from deep sea cores and benthic stacks. And what's the large-scale pattern that we observe? So all of you are familiar with this, but basically we have a long-term cooling process over time, which is reflecting a drawdown in CO2 in the atmosphere over this long-term period. And as temperatures cool, we see first the onset of southern hemisphere glaciations. There's kind of a brief warm spell on the early Miocene, followed by then the further cooling, the onset of northern hemisphere glaciations. And what's happening in a global sense is we're actually removing energy from the climate system, which means we have less rainfall overall, and that favors certain kinds of primary producers of our others in terrestrial ecosystem. So if we look at, for instance, grassland habitats, the first grass fossils actually appear prior to the KP boundary, but we see the emergence of large-scale grassland habitats. These are C3 grasses, first in the Eocene and Oligocene, and not very long afterwards, we see herbivores moving onto those landscapes and exploiting them. There's the famous record, for instance, of horse evolution that you see in the Americas and Eurasias. And then later, as temperatures further cool and landscapes dry, and CO2 declines, you see the emergence of these special arid-adapted and low CO2-adapted C4 ecosystems, which you'll be familiar with probably as well, with animals moving into those too. This happens in the Americas, in Eurasia, and ultimately in Africa. And this is really important, this large-scale process, in terms of thinking about the origins of apes and then the hominins. It's actually during the early Miocene, latest Oligocene and early Miocene, that you see the radiation of large-bodied apes in Africa. And we have traditionally thought that that has happened in a relatively well-forested and perennially wet ecosystem on that continent. And you see quite a radiation diversity of specimens, which then move into Eurasia. And then in the aftermath of that process, as really conditions dry substantially in Africa, you see the origin and adaptive radiation of a whole series of different hominin species. And this is, of course, important to our own ancestry. Now, some of this kind of canonical information has been challenged by a pair of papers that have come out in the journal Science in the last several years, which are really interesting. This is work led by an African Miocene research team called the REACH Project. Daniel Pepe, Susie Cody, Kieran McNulty have really spearheaded this important work. And if you look at a variety of terrestrial records, what you see is that if you can examine, for instance, soil organic matter, you can look at plant waxes, you can look at pedogenic carbonates, and the heavy carbon contribution, so heavy carbon isotope composition and all of these different proxies indicate that far earlier than the latest Miocene and Pleistocene, you actually see what seems to be some spread of C4 grasses. And this is not just a small amount, but as the authors indicate, kind of actually abundant C4 grassland habitats. You can use the plant wax record, for instance, to try to calculate some maximum percentages of total C4. And so the authors estimate that in some places we may have as much as 60, 70, 80% C4. And this is really rewriting the story of human and ape origins. And it indicates that this proliferation of large-bodied apes may be occurring not in a forested landscape, but instead in an open landscape, which leads us to ask, well, what is really special about climate in the Pleistocene that leads to the emergence of hominins? And I should just mention, all these data are being taken from the East African Rift Valley, which I'm showing you here in NASA. So I apologize. I know that most of you learned all of this in like elementary school, but it's worthwhile just mentioning quickly again. So there are two stable isotopes of carbon, carbon-13 having an extra neutron. Now, when plants using C3 photosynthesis, which is the most common form of photosynthesis on Earth, are capturing CO2 and carbon from CO2 in the atmosphere, there's this enzyme rubisco that's grabbing the CO2 and putting it ultimately through the Calvin cycle into sugars. And rubisco discriminates against heavy carbon. So as a result, carbon that you find in sugars tend to be depleted in delta 13C compared to what in 13C compared to what you would find in the atmosphere. Now, if CO2 concentrations become low enough in the atmosphere, actually sometimes you can have oxygen binding into this system instead of CO2, which causes a very costly phenomenon, photorespiration. Actually, about 30% of terrestrial plant productivity is lost to photorespiration, and this is especially important in the later Cenozoic as CO2 concentrations decline. So again, I know you all know this, but it's worthwhile just mentioning one more time. Plants devise a very ingenious strategy, many times independently, actually, to deal with this problem. They separate the process of carbon capture and then incorporation of the sugars into two different bundles. And the outcome of that, and I won't go into why because we're sort of running a little bit late on time, is that you see more of the heavy carbon in plants that are especially adapted to dry environments and low CO2 environments. We call these C4 plants. So that's the carbon record. And then to look at oxygen for a moment, again, oxygen isotopes are non-randomly distributed on Earth and actually throughout the universe. On Earth, this is based upon mostly the hydrological cycle. So you can imagine if you have some water body, let's take the global ocean, for instance. Sunlight is arriving at the surface of the ocean. You have the water-gas interface, and some water molecules are evaporating. Water molecules that have the light isotopes are just statistically a little bit more likely to go through that liquid-to-gas transformation. And so the exact opposite happens when you have rainout. But the important consequence of this is one that, for instance, if you're in the tropics, during dry seasons, you tend to see enrichment in the heavy oxygen isotope, so more delta 13 or delta 18O. And during wet seasons, you tend to see less of it. And just more importantly, is in a general sense, the oxygen isotope composition that you measure in waters or in animals' bodies who are drinking those waters, eating plants that depend upon those waters, they reflect the larger hydrological system on this landscape. Now, of course, these changes are transient. We don't even know what rainfall was like last week or last year, unless we're taking some kind of record. But it turns out that animals are taking this chemistry from their environment, incorporating it sequentially into hard tissues, including teeth. And in hydroxyapatite mineral, there are multiple molecular species that take carbon, one that takes carbon, several that take oxygen. And so these transient signals can be recorded into hard tissues. And teeth are especially useful because they form in layers. So in this project, the trichomycin project that I was associated with, collected in the end hundreds of dental specimens from the East African Rift Valley to try to see, okay, we see these kind of records from organic matter and soil, from plant waxes of C4. How is this getting incorporated into diets of animals who are living on these landscapes? So we're going to use teeth to look at these records. The first thing that we see, if we look at oxygen isotopes, you can see a blue line there in the background moving from the Oligocene of the bottom towards the present at the top. This is that Westerholt or Zachos curve showing us what's happening in the global oceans. And as time passes, the Earth cools and dries, you see that values become higher over time. And actually, the exact same process is happening in a general sense in the terrestrial records in East Africa as well, from herbivore teeth. And so what this shows us is that landscapes are just drying out over time. Something which is important is that you see that some of these very high values of delta 18O are actually rising faster than what we see in the benthic record, which indicates that some amount of drying is happening even more quickly in this region than we see kind of in a global sense. These are, of course, totally different properties, but there is a similarity of pattern. Now, really curiously, and this is something that we're struggling with, it's really not until the latest Miocene that we see any incorporation of C4 resources into herbivore diets. And that's perplexing because we have all of these records from sediments, essentially, telling us that there should be abundant C4 on these landscapes. And we see almost none of it being incorporated into herbivore diets. And there's a lot of papers that from this region and other places too, indicating that when resources are available, there's maybe some amount of lag in terms of when herbivores begin using these resources, but those lags tend to be on the order of hundreds of thousands or maybe a few million years. So waiting for a 10 million year period is sort of surprising in some way. And we don't fully know why this is the case. It could be a problem with the dental record, for instance, and maybe there's some reason why C4 resources are not getting into these specific teeth, or maybe are diagenetically being removed in some way. Another possibility is that diagenesis may be preferentially affecting soil organic matter or reflexes, carbonates as well. So this is something that we're really actively exploring right now. If we look at that record in more detail over time, actually one of the earliest sites that we explore is a site called Topernawi. So this is from the eastern side of Lake Turkana in northern Kenya in the middle of the Rift Valley. It's really interesting actually as a snapshot of the world that existed in Africa before the African continent collides with Eurasia. And you have a land bridge that forms, bringing us many of the modern African fauna that you think of today when you go on safari, for instance. But at this time, most of the world and the fauna in Africa are quite different. We have some proboscideans. We have a few primates, which, as they love to do, have rafted over earlier than everybody else. And the vast majority of what we find are hyraxes. Actually, we had estimated early on that about 85% of the specimens that we found from the site were hyraxes. And after a very, very painstaking faunal analysis from several experts, it turned out that it's over 90%. So some of our fancier tags have converted into hyraxes. So it's a very different kind of world. But if you look at the oxygen and carbon isotope space, you can see we're in this bottom corner here. And that just means that we're very firmly in a C3 forest. And we're in a wet, perennially wet environment. So it's not like the African landscape that we see today. And if you move forward in time, however, you can see that we begin creeping up towards higher values of oxygen isotope compositions. And this is indicating that these landscapes are drying. And we're pushing up against the boundary of this C3 to C4, at least mixed C3, C4 transition, but we're really staying in the C3 world. But you can see that despite the fact that we're staying in the C3 world, there's a great amount of hydrological variability with some places being far drier than others. And we're seeing this, each of these dots is an individual animal, and each of these colors is one site that we're looking at. So you can see there's a kind of really substantial spread of data. It's not then until we move into the latest Miocene and Pleistocene, when we see the emergence of hominins, that we move very, very far into this mixed C3, C4, and then C4 world. And this is going all the way then to into Lothicum, a famous site also from this region, with the appearance of an Australopithecines. So that's really interesting to us because it does show us that geochemically and in terms of primary productivity, hominins are emerging in a kind of unique landscape, but African apes, who are their precursors, are somehow living in an area, in an environment which is not purely forested or wooded. There's actually some mix of probably open habitats as well. And we're not seeing any evidence of C4 grasslands, which Pepe and Kieran McNulty have found, for instance, but we are similarly finding evidence of at least hydrological diversity, which would support some aspect of their claims. Now we can zoom in on any one of these sites to try to see what's happening at a given location. And Locheringen is a really interesting site because we do have some large-bodied hominoids. These are Afropithecus specimens, generally the first large-bodied apes that we find in the world and in this region, kind of precursors to chimpanzees, gorillas, and hominins. And we have a whole range of taxa. If you look at their oxygen isotope compositions, you'll see that some things are exactly as you'd expect. So for instance, the arachnoids all the way on the right have high values. They're browsing from canopies. You can see that the anthracotheres, which are, with some small amount of controversy, considered likely to be hippopotamus precursors. So they're spending a lot of time in standing water bodies, terrestrial water sources. Their values are very low. And that's what you see in modern giraffoids and modern hippos today. And that's just because water from leaves tends to be very evaporated. Here you can see those kind of leaves are small engines of evaporation for oxygen isotopes that are enriching the heavy isotope. So animals like giraffes that derive a lot of their water, either liquid water from leaves or maybe metabolically produced water, tend to have higher values. And the Afropithecus specimens from this site have high values, which is what you see in modern primates today who live in canopies. Chimpanzees are canopy primates as well. So everything is expected. Now, if we go to the other side of Lake Turkana, really from the same age, basically, this is a site Bullock, which was discovered not very long ago. It forms during the Middle Miocene climatic optimum. You see some very similar things. So for instance, our giraffids and anthracotheres are on either side of this oxygen isotope spectrum, indicating that we have browsing from canopies, we have animals living inside these standing water bodies. But really interesting for us, the primates, including the Afropithecus specimens, are down next to the anthracotheres. And this is really perplexing. We see it for quite a few specimens, and it's not something that we see at any other site from this time period. It's not something that we see in other primate species today. And we're curious about why this is the case. We have some theories about it. One thing that we believe may be the case is that if you compare the aridity at Bullock, which we can estimate in a mechanism I'll explain in a moment, compared to other sites, aridity at Bullock seems to be a little bit higher. So this is just calculating the total amount of rainfall which is arriving on a landscape and comparing it to the amount of evaporation occurring on that landscape as well. And there seems to be just more evaporation happening at Bullock compared to other places. This R1, R2, R3, R4, R5, these are different calculations of what's called the aridity index. And what we do is we actually subtract the oxygen isotope compositions in hippos from what we observe in giraffes. And I won't explain why, but effectively, if you plug in the equation hippo minus giraffe, it gives you something like aridity. And there's a couple of really great papers on this topic. Scott Blumenthal has published a recent one, which we're using here. But this suggests to us that there's something about the water which is present at Bullock, which is causing primates at that site to make different behavioral decisions about where they access their water, maybe seasonally or even year-round. And this is interesting for us because we know that today among modern African apes, behavioral diversity is really extraordinary, both in terms of the kinds of foods which are consumed over the course of seasons, but also how water is consumed as well. And so knowing that that was occurring 17 million years ago is fascinating, and it leads us to think about what kind of behavioral complexity existed long before the emergence of hominins on at least partially open landscape. So what if we try to delve in a little bit further to look at behavioral diversity among these Afropithecus specimens, which are interesting because they're so large and they have very thick enamel as well, and are thought to be potential precursors of modern African apes. So first of all, I'll just say that there is a long and wonderful history of the exploration of seasonal patterns of ecology in the study of evolutionary biology. Darwin actually writes about it extensively. I don't know if you have a chance and you haven't in a while to look at chapters 3 and 4 in particular. He also goes into this in chapters, I think it's 11 or 10 and 11, but now it's been enough time. I should look at those again as well. But Darwin has a lot of really wonderful commentary on this topic in this book. And there's still a lot of ongoing research today. So, for instance, it's known that some animals use tools and do so seasonally. This is true for all kinds of primates from Asia and Africa all the way to South America. One example that I love is actually finches from the Galapagos Islands. Some finches use, these are cactus finches, they use sticks to forage for grubs. And they do so only during dry seasons. And the ones that do so more than others actually have higher reproductive success, which I found just to be really interesting. We also see actually that seasonality of rainfall and green up or the production of new plant material can pattern foraging behaviors and even speciation. There was a recent analysis of the genomic data from different giraffe subspecies across East Africa. And the genomes revealed that actually there's very little gene flow between these different subspecies, which led the authors to conclude that they're really proper species in their own right. Their distributions are almost perfectly aligned with green up and the arrival of the range with the intertropical convergence zones across this larger landscape. And interestingly, this radiation of giraffes is happening around 2.5–3 million years ago, which is exactly when we see hominids radiating as well. So seasonality can drive behavior and also reproductive isolation. There has been a very long and wonderful commentary on the potential role of seasonal ecology and water availability in driving hominid and primate evolution. This is a paper from Peter Andrews and Lawrence Martin arguing, using Afropithecus, for instance, that it's possible that dry forests may have driven special aspects of thick enamel and ape ecology in the early Miocene. And this was a very interesting paper because this came out long before we had any evidence that from the terrestrial and geological record that this area may have been more arid in the early Miocene. This pair of papers that came out in Science also make a similar case from a site in Uganda, actually, arguing that Moritopithecus, another somewhat larger ape, not quite as big as Afropithecus, had some amount of behavioral versatility, so may have been moving on the ground in some between forest patches, for instance. And Aaron Westling has published a lot of just spectacular papers on the role of savanna environments in modern chimpanzee ecology as well, from Tanzania to Guinea. So there's a really wonderful commentary on this. And I'll just try to make a case now that teeth can tell us a lot about seasonal ecology as well. Now, why is that? So teeth are largely mineral, so about 90–95%, enamel at least is 90–95% mineral. But there's an important organic component. And even though in the mature teeth, that organic component is fairly small, this organic component plays a crucial role in driving the specific ordered pattern of tooth formation, which allows it to achieve those high mineral densities, and also, as I'll show, can help us achieve a temporal record. So what I've done in this study, this is a small study in proteomics that I conducted a few years ago with colleagues at the Forsyth Institute in Boston. It's a medical dental kind of research facility. We took a series of teeth that were still forming in utero, teeth that were then erupting. And from the bottom of the teeth forming in utero towards the top of the teeth, which is erupted, you kind of get a sequence of tooth formation from initial secretion of a mineral protein water matrix to then a maturation of that matrix, then to a final eruption into the jaw. And if you take any small parcel of these teeth from these different zones and you examine them for the proteomic composition, you can see, for instance, on a principal components analysis, that the secretory locations cluster quite specifically in terms of how they express proteins, what proteins they express and in what abundance, compared to maturing enamel or then later enamel that has erupted. We can contrast also those results with dentin or bone or other kinds of tissues. One thing we see is that the total amount of protein in the secretory region of the tooth, and you can see this right here, sorry, people on Zoom, you can't see me pointing, but we find the most protein, of course, where the matrix has just been deposited. So actually this protein matrix is really guiding tooth formation. And when the matrix is first formed, most of what you have is either water or protein. But then as time passes, these proteins are removed. And then you grade toward the kind of mature and highly dense mineral. Actually, the highest diversity of proteins that you find is in the maturation phase. That's just because you tend to keep some of the secretion secretory proteins in that phase, but then you have new proteins which are emerging as well in the maturation process. Um, when we do this kind of study, we find amyloblastins, enamelins, amylogenins, and different isoforms which are the structural proteins that we expect to find inside a mineralizing tooth. This is um, long-standing proteins which have been studied to understand the tooth formation process from a biomedical perspective. We also find a series of proteolytic enzymes that help remove these during the maturation process. And we find these proteins exactly where we expect them in the formation process. Um, what is important is that all of these structural proteins guide a sequential layer by layer formation of the mineral then. And if we sample across these layers, which are guided by the formation by these proteins, using something like a laser system or maybe an ion microprobe, we can make measurements of, for instance, oxygen isotopes, which will give us a record of seasonally fluctuating body chemistry records, which are representing what's happening in some transformation system in the landscape. And especially if we look at those layers under our microscope and align the layers with the measurements we've made, we can day by day basically track what's happening inside these teeth over time. So if we look at modern specimens, we can take a pair of baboons that died not too long ago from the Awash, where we find a lot of also fossils today. And we know when these individuals died. We have rainfall records from when they died. Going upstream, we can see that from Addis, a little bit in the highlands just above them, when the rains arrive in blue on the right there, in July and August, oxygen isotope values decline. So heavy oxygen isotope values decline. And this is just showing us that hydrologically, oxygen isotopes are tracking what's happening in the landscape exactly as we expect from wet seasons to dry seasons. And then if we look at in black over there, next to the little cartoons of those two individuals, we look at in black at the fluctuations in their teeth compared to the rainfall which is present in the landscape, that there's an inverse relationship between what we see in their teeth and the rainfall in the landscape. And again, this is important. It's kind of demonstrating that there is some relationship which is expected again between what we see in teeth and what's happening hydrologically in the landscape. One thing which I'll just mention is actually these two individuals come from two different troops in Awash, which are drinking from two different water sources. One is drinking from a shallow lake, the one up above, Lake Bazaka. The second is drinking from the Awash River, which is dammed and has about a five-fold volume compared to Lake Bazaka. And you can see that the fluctuations are much more extreme and responsive to rainfall patterns in Lake Bazaka compared to the individual drinking from the Awash River, which we think is just a mass balance question about how fast can a water body respond to wet and dry seasons if it is fairly shallow, for instance, as opposed to dammed and kind of slowing down these cycles over time. So the source of water, even on a local scale, does matter. We can look at the same pattern on the other side of Africa. These are chimpanzees from Liberia. You can see that there's a very nice relationship between oxygen isotopes measured from their teeth and rainfall patterns on these landscapes. These were collected by a scholar named Harvey for Harvard back in the 1930s under conditions that were somewhat dubious. Some of my students have been looking into that recently. But anyway, climatologically, at least, you can see there's a very nice correlation, which is helpful. And to be honest, we can look at primates taken from all across the African tropics, and you see this kind of very nice relationship between oxygen isotopes and rainfall, and what you find in enamel as well. So what if we look at Afropithecus, for instance? Now, these are two specimens taken from a site called Caladir, also 16, 17 million years old, also from Lake Turkana. We have two specimens that are separated by about 40 meters of sediment. And what you see is, yes, we do have these in blue. They're pronounced in somewhat extreme wet seasons, but they're separated by prolonged dry seasons, which are kind of interesting. And the individual who's a little bit younger, taken from higher up in the section, you can see that those oscillations are somewhat more pronounced, so higher amplitude variation, and they're a little bit more irregular. The individual who's a little bit older has more pronounced annual cyclicity, but it's really dampened variation. And something which I find fascinating is that actually the overlap of these values is, there's almost no overlap whatsoever, which is kind of interesting. So the same individuals, the same species, sorry, the same section, the same site, but you can see that the quite different hydrological regimes. And we're very curious about this. I know that some of you are working from from deep sea cores. We ran Lomscargle periodograms, or basically Fourier transformations on these signals, and we do this with all of our teeth now. And we can see that there's strong annual cyclicity in both of these records. You see this on the left here with like that larger blue dot. But you can see that in some of the individuals, the younger individual, actually subannual cyclicity is stronger. And so we don't quite know why this is the case, but we teamed up for this project with a climatological modeling group. They're using community Earth system models. This is Chris Polson and his graduate student Diane Lee, who's actually defending later today at the University of Michigan. And they showed that if you actually slightly alter Milankovitch cycles, or kind of parameters of the way that the Earth rotates around the sun, the tilt in this case, you can generate either higher or lower amplitude rainfall patterns and shift from unimodal to bimodal rainfall patterns, which we thought was very interesting. And this may explain why we see such different patterns of seasonal variation at these two different locations in the section. And one thing that we take away from this is that not only were these primates dealing with prolonged dry seasons at these sites, but also vastly different climatological regimes across Milankovitch cycles, which won't surprise any of you who work in that kind of space, but something important for us to keep in mind in terms of the behavioral flexibility of these individuals living on these landscapes. One other thing that's really useful to think about when we have this kind of multiple measurements with high resolution through ion microprobe of these individual teeth, we can get a lot of data that you miss through traditional drilling methods. So here you can see over 60 different individuals from the same population in Liberia, from which I showed you these chimpanzees just a moment ago. And if we sample these 60 different individuals, we get a spread in oxygen isotope composition of about 2.7 per mil. So that's not bad, actually. But then if we take just one of these individuals, which I'm showing here in blue, and we measured two of them, one of them was not from that original sample, we have variation of nearly 9 per mil. So what that's showing us is that from a behavioral, well, from a geochemical, obviously, but also from a behavioral perspective, there is a massive amount that we're losing when we do this conventional drilling. And if we actually want to understand behavioral variability, we really should be using these higher resolution methods, because where these animals are getting their water from and how they're feeding over the course of a year changes dramatically. We can look at the same thing with our two Afrobithecus specimens. These are the two that we measured. Here are 101 hominins measured through conventional methods over the last, over a 4 million year period. And you can see that we have the same range of variability from two specimens that we measured using this high resolution method compared to all the data collected from all hominins basically in the same region, writ large, over 4 million years. So we really need to be moving more towards these kinds of methods to get at this behavioral variability. You can do more than just measure these squiggles in these teeth to try to understand where they come from. There are a pair of landmark studies that were published by Benjamin Passy and Terry Serling 2 decades ago, actually now, and this is when the last paper was published. And what they do is they establish a model of how a tooth actually mineralizes over space and time, and then uses, in this particular case, the linear algebra to solve from measurements that you take in the teeth of these individuals for the original variation in body chemistry over time. And I'm also very interested in this topic. I've used a very similar kind of methodology, but instead of linear algebra, this particular paper used a Bayesian inverse system. But the idea is that you have some kind of landscape where you have a seasonal rainfall pattern, and I'm showing this here at the top there. And animals living on that landscape is drinking water, eating food. It grows its tooth. You take measurements from the tooth. The measurements with these in black circles there don't really look anything like what the environment that the animal actually lived in. And that's because the teeth are not simply forming in layers. It's actually a multi-wave process of formation, which is dampening and distorting the original seasonal signal. But if you have a model of how that mineralization process works, you can make a mathematical kind of hypothesis, which is that the probability of a given drinking water history or climatological signal on the landscape, given the data that you've measured, given your models of tooth mineralization, and given some appreciation of error and maybe a few other priors, is equal to the product of a series of likelihoods, which are actually, then you flip the mathematical proposition. The likelihoods are the likelihood of measuring that data given this hypothetical drinking water history. And it's practically very difficult to solve that equation. But if you iteratively propose drinking water histories and solve them one by one, on a computer, you can do this millions of times. And actually, this method was originally developed during the Manhattan Project, trying to figure out the capture area of nuclei to understand how uranium atoms might absorb neutrons and then engage in fission. You can calculate these probabilities very, very quickly, so tens of thousands of times, hundreds of thousands of times, and derive a most likely probability of drinking water history, which resembles much more closely the original pattern or seasonal pattern that the individual experienced compared to what you actually measure from the tooth. This seems complicated, but it's actually not that bad. And I've been developing a code which we're hoping to publish in the next year, which puts all these methods online, both a linear algebra method originally developed by Ben Passy and Terry Sterling, and also this Bayesian inverse system. And nobody has to download any software on the computer or even understand how it works. All you have to do is input a little bit of information about the taxon and the tooth that you're looking at, and the data, of course, that you measure from the tooth, and you can get all of this directly. It's actually run on the cloud, so you don't have to have anything happening on your own computer. This model takes into account certain characteristic aspects of tooth formation. I'll skip over this for the moment in the interest of time. It models in bright yellow there, and then the secretory process, and then later in green, kind of maturation process later, how a tooth forms over time. And you can see a kind of video of this here, this highly tunable model. This is work that I have been doing with one of the graduate students of Thomas Tutkin up in Mainz, where I'm headed soon. And these are horses from the Eemian, and we get really spectacular, beautiful, highly annual records from this site looking at horses. And you can see in kind of stars are the original sample values and what we reconstruct in blue. So anyway, we're very excited about these methods. I'll just mention actually a postdoc who I've been working with recently, who's at Utah, just a couple days ago published a paper using some of these methods, so I recommend this. It's going to make a plug for this paper in case you are interested. And also, of course, we're interested in the radiation of hominins. So what about applying this to hominins as well? There's a really cool paper that came out recently by Kerry Mongol, Louise Leakey, Mead Leakey, and others. Cyprian Niete have led a lot of the excavations, finding this incredible Paranthropus hand at the Kubi-Fora region of Turkana. And actually, we've been to that very site, just not too far from it. We have literally a Paranthropus and a Homo who are from the exact same depositional layer, only a few meters from each other. We have their teeth, and we've been using these same inverse methods and high resolution measurements on those specimens and a whole series of others in hominins. And I have not put the data into this talk, but if you are interested, please come talk to me about it afterwards. But what I will say is that we see really dramatic differences in how these two species, who it turns out are not necessarily, we used to think that, okay, Homo was making tools and Paranthropus and Australopithecus were not, and that's now really in doubt. But what distinguishes these two different taxa? And they're using seasonal water resources extremely differently, which is really fascinating for us. Part of the reason that we can explore these issues of seasonality is because of this kind of interesting process of tooth formation involving a mineral and protein interaction. We can actually look at the proteins themselves. Besides hominids, there's all these other interesting animals on these landscapes. We have dinotheres, which are now extinct. We have anthracotheres, whose relationship to hippos is not fully understood. We have these erythropods. Their entire order is extinct. We think that they're Afrotherians, but that's not fully known. All these taxa from the ancient African world, which no longer exist. But what if their teeth preserve some of these proteins that help guide the formation process, which is so useful for studying geochemistry, for instance? What if you look at the organic phase? So much is contained within these teeth, deeply embedded in modern teeth, and some work has now been done pushing the discovery of proteins in teeth back into the Pliocene. What if we can go back even further? So we've been looking at this. And before I go there, I'll just mention, so I mentioned, I think, ameloblastin, amelogenin, enamel, and a few structural proteins which are well known in teeth. But actually, from our proteomic analyses of modern teeth, we find hundreds of teeth which are very poorly studied. And we find the classical proteins that everybody expects, and we find many that are not expected. And just to give you an example of some of these, neurotensin, we know that it's involved in neural signaling. It actually has been found using other methods before in rat enamel organs, not in enamel itself, but enamel organs. If you don't have it, as a human or a mouse, you have weak enamel, basically, so enamel hypoplasia is demineralized enamel. So we know that it's somehow medically critical for the formation of healthy enamel. It's never been found in enamel before, but we find it using proteomic methods, and it's doing something that's crucial to make healthy enamel. Another protein, laminin A3, this is involved in supporting junctions between cells. It has been observed in complexes during tooth bud formation. And also it's actually been observed at the interface in ameloblast between enamel, never in enamel itself again. But if you don't have a functional copy of laminin A3, your tooth will have pits and grooves and demineralized zones as well. So somehow important, never observed directly in enamel before, but we're measuring it there and it's important for formation. My favorite protein that we find is alpha casein S1. So again, never found in enamel before, but it's a major constituent of breast milk. And this is really interesting because, okay, for one, it's part of the secreted calcium phosphoprotein family. So an ancient family of proteins that are important in hard tissue formation in many different vertebrates and other taxa. We know that alpha casein S1 can stabilize amorphous calcium phosphate at high solute concentrations, and that's one of the reasons why it's probably in breast milk, is that it's like basically facilitating the delivery of calcium and phosphate to infants. But also, it's actually been used recently by bioengineers to try to make new enamel mineral material, and we're finding it in secretory enamel, exactly in the area where we actually know that there is amorphous calcium phosphate in developing teeth. So anyway, with that as a precursor, we went to some of the teeth in the fossil record going back 30 million years. And we examined them for proteins. And what we basically find is that as time passes, you find proteins and they degrade. So you find less and less over time. But we find them even in the oldest specimens. Here's an example of the some of the mass over charge peaks that we find for a series of different fragments from, actually this is a dentin protein, which has been found in enamel as well. You can see that above what we measure is exactly what we predict below, using modern, basically neural network methods designed to predict what you should observe from the spectrum. And what we can see is that over time, the amount of material that we find in these teeth degrades, but we're still finding small components even after quite a bit of time has passed, going into the early Miocene and even Oligocene. We can use these kinds of sequences to actually try to reconstruct phylogenies for extinct taxa. And we're finding only small quantities of material, but we're hopeful that with methods developing every single year, the abundance and length of these sequences is going to increase, and these methods of reconstructing phylogeny will improve. And I should say, actually, the day that our paper came out in Nature, another paper was published finding similar results from the Arctic, which we're very excited about. And their preservation is even better than ours, as is expected given the temperature history of both locations. So we have a few next steps. We're interested in the pattern of decay for these specimens. We're doing some experimentation to see basically how these materials degrade. And some of this is actually just by looking at fossils that are 100,000 years old, 500,000 years old, 1 million years old, and so forth, to try to really test how degradation patterns look going back in time more methodically. I'm actually hosting a workshop in Kenya. We're bringing people from Copenhagen and all kinds of places to Cambridge to talk about this summer. So if you know anybody who's very interested in proteomics, please let me know. I want to say one last thing before I sign off, which is that actually my grandmother, who I was very close with before she died in 2001, was born in Berlin. She spent a lot of time in the city. And so I just want to thank you for letting me come and give a talk here, because she loved Vienna. In fact, she sang several songs in New York and throughout Europe after the World War II about Vienna, and kind of harking back to the late 20s and 30s, a period of her kind of youth. She was an operetta and cabaret singer. Her father was the first conductor of the Berlin Opera House in Germany. And she was friends with Bertolt Brecht, Kurt Weill, George Grosz. Her father played in a string quartet with Albert Einstein in Berlin. So I'm just really grateful that you're allowing me to come and also be in the city where she spent quite a bit of time in. So with that, I'd love to take questions.